The intricate relationship between sleep and brain health has captivated neuroscientists for decades, but recent breakthrough research from Washington University School of Medicine has unveiled a potentially transformative discovery that could reshape our approach to treating devastating neurodegenerative diseases. A medication commonly prescribed for insomnia has demonstrated remarkable protective effects against the brain damage characteristic of Alzheimer's disease and related disorders, offering new hope in the fight against these progressive conditions that affect millions worldwide. The convergence of sleep science and neurodegeneration research. For years, researchers have observed that individuals who experience chronic sleep disturbances face an elevated risk of developing Alzheimer's disease later in life. This connection, while well documented, remained poorly understood at the molecular level until relatively recently. The latest findings from Washington University represent a crucial advancement in our understanding of how sleep quality directly influences the accumulation of harmful proteins in the brain, particularly a protein called tau that plays a central role in multiple neurodegenerative conditions. The research team, led by distinguished neurologist David M. Holtzman, has been at the forefront of investigating the sleep neurodegeneration connection for many years. Their previous work established fundamental links between sleep deprivation and the accelerated accumulation of proteins associated with Alzheimer's disease. This foundation set the stage for their current investigation into whether improving sleep quality through pharmaceutical intervention could potentially slow or prevent the neurological damage these proteins cause. The medication at the center of this groundbreaking study, Lemborexant, belongs to a relatively new class of sleep aids that work through a different mechanism than traditional sleeping pills. Rather than broadly suppressing brain activity as many older sleep medications do, Lemborexant specifically targets the orexin system, a network of neurons and receptors that plays a crucial role in regulating wakefulness and sleep cycles. This targeted approach not only promotes more natural sleep patterns but, as the new research reveals, may also confer significant neuroprotective benefits. Understanding the tau protein and its role in brain disease. To fully appreciate the significance of these findings, it's essential to understand the role of tau protein in both healthy brain function and disease. Under normal circumstances, tau serves vital functions in neurons, helping to maintain their structural integrity and facilitating the transport of nutrients and other essential molecules along the cell's internal highways. The protein achieves this through a delicate balance of chemical modifications, particularly the addition and removal of phosphate groups that regulate its activity. However, in various neurodegenerative diseases, this careful balance becomes disrupted. Tau proteins become excessively decorated with phosphate groups, a process known as hyperphosphorylation. When this occurs, tau proteins lose their normal function and begin to clump together, forming tangles within neurons. These tangles not only impair the neuron's ability to function properly but also trigger inflammatory responses that ultimately lead to cell death. The progressive accumulation of these tau tangles correlates closely with cognitive decline and the severity of symptoms in Alzheimer's disease and several other neurodegenerative conditions. The distribution and progression of tau pathology in the brain follow predictable patterns that researchers have mapped extensively. In Alzheimer's disease, tau tangles typically begin forming in brain regions critical for memory formation, particularly the hippocampus and entorhinal cortex, before spreading to other areas as the disease advances. This pattern of spread corresponds closely with the progression of symptoms, from early memory problems to more widespread cognitive and functional impairments. The innovative experimental approach. The Washington University research team employed sophisticated experimental methods to investigate Lemborexant's potential neuroprotective effects. They utilized genetically modified mice that developed tau pathology similar to that seen in human neurodegenerative diseases. These animal models provide invaluable insights into disease mechanisms and potential therapeutic interventions, allowing researchers to study the progression of tau accumulation and its consequences in controlled conditions. The study design compared several groups of mice, those receiving Lemborexant treatment, control groups receiving no treatment, and comparison groups receiving zolpidem, a widely prescribed sleep medication from an older class of drugs. This comparative approach allowed the researchers to determine whether the observed benefits were specific to Lemborexant's mechanism of action or simply a result of improved sleep regardless of the medication used. Over the course of the experiment, the researchers meticulously monitored multiple indicators of brain health and disease progression. They measured the volume of key brain structures, assessed the accumulation of abnormal tau protein, evaluated markers of inflammation, and quantified the extent of neuronal death. These comprehensive assessments provided a detailed picture of how Lemborexant treatment influenced the trajectory of neurodegeneration. Remarkable findings and their implications. The results of this investigation exceeded expectations in several ways. Mice treated with Lemborexant showed substantially less tau accumulation compared to untreated controls, with particularly striking preservation of brain structure. The hippocampus, a brain region essential for memory formation and one of the first areas affected in Alzheimer's disease, maintained significantly greater volume in Lemborexant-treated mice. The preservation represented approximately 30 to 40% greater volume compared to control animals, a difference that could translate to meaningful functional benefits. Perhaps most importantly, the study revealed that not all sleep improvements are created equal when it comes to neuroprotection. While zolpidem successfully increased sleep duration in the experimental mice, it failed to provide any of the protective effects against tau accumulation observed with Lemborexant. This critical finding suggests that the mechanism by which sleep is improved matters tremendously for achieving neuroprotective benefits. The specific action of Lemborexant on orexin receptors appears to be key to its therapeutic potential. The research also uncovered important details about how Lemborexant exerts its protective effects at the molecular level. By blocking orexin receptors, the medication appears to prevent the excessive addition of phosphate groups to tau proteins, maintaining them in a healthier state. This mechanism suggests that orexin signaling may play a previously underappreciated role in regulating tau phosphorylation and that targeting this system could represent a novel therapeutic strategy for neurodegenerative diseases. Sex differences and unanswered questions. One unexpected finding that emerged from the study was the observation that Lemborexant's neuroprotective effects were apparent only in male mice. This sex-specific response raises important questions about the biological factors that might influence treatment efficacy and highlights the complexity of developing therapies for neurodegenerative diseases. The researchers noted that female mice in their experimental model developed less severe neurodegeneration overall, which might have made it more difficult to detect protective effects of the treatment. This observation underscores the importance of considering sex as a biological variable in neuroscience research and drug development. Hormonal differences, variations in gene expression, and distinct patterns of brain aging between males and females could all contribute to differential responses to therapeutic interventions. Understanding these differences will be crucial for developing personalized treatment approaches that can benefit all patients affected by neurodegenerative diseases. The research team continues to investigate the mechanisms underlying these sex differences, work that could reveal important insights into the fundamental biology of neurodegeneration and potentially identify additional therapeutic targets. This ongoing investigation exemplifies the iterative nature of scientific discovery, where unexpected findings often lead to new questions and deeper understanding. The broader therapeutic landscape, the potential applications of these findings extend well beyond Alzheimer's disease alone. Abnormal tau accumulation occurs in multiple neurodegenerative conditions, collectively known as tauopathies. These include progressive supranuclear palsy, a disorder affecting movement and balance, corticobasal syndrome, which causes problems with movement and cognition, and certain forms of frontotemporal dementia, which primarily affect personality and language. The possibility that Lemborexant or similar medications could provide benefits across this spectrum of diseases represents a significant opportunity to address currently untreatable conditions. The timing of this discovery is particularly fortuitous given recent developments in Alzheimer's disease treatment. While newly approved antibody therapies targeting amyloid protein have shown modest benefits in early-stage Alzheimer's disease, many experts believe that truly effective treatment will require addressing multiple disease mechanisms simultaneously. The ability to target tau pathology through a well-tolerated oral medication that also improves sleep quality could complement existing and emerging therapies, potentially leading to more substantial clinical benefits. Moreover, the preventive potential of this approach deserves serious consideration. Given that pathological changes in the brain begin years or even decades before clinical symptoms appear, interventions that can slow or prevent the accumulation of harmful proteins could have profound implications for public health. A medication that people could take to improve their sleep while simultaneously protecting their brain health represents an appealing preventive strategy, particularly for individuals at high risk for neurodegenerative diseases. Clinical translation and future directions. The translation of these promising preclinical findings to human patients will require careful planning and execution. Lemborexant already has regulatory approval for treating insomnia, which could potentially accelerate its evaluation for neuroprotective indications. The established safety profile of the medication in humans provides valuable reassurance as researchers design clinical trials to test its efficacy against neurodegeneration. Several key questions must be addressed through human studies. First, researchers need to determine whether the tau-reducing effects observed in mice translate to humans with neurodegenerative diseases or those at risk for developing them. This will require sophisticated imaging techniques and biomarker assessments to measure tau accumulation and brain health over time. Second, the optimal timing, dosing, and duration of treatment need to be established. Should Lemborexant be used preventively in high-risk individuals, or would it be more appropriate for those showing early signs of cognitive decline? The research team at Washington University, in collaboration with pharmaceutical partner Eisai, is already planning follow-up studies to address these questions. These investigations will likely include both observational studies examining brain health in people already taking Lemborexant for insomnia and controlled trials specifically designed to test its neuroprotective effects. The involvement of a pharmaceutical company with expertise in drug development and clinical trials could help accelerate the translation of these findings to patient care. The importance of sleep in brain health. This research adds to a growing body of evidence highlighting the critical importance of sleep for maintaining brain health throughout life. During sleep, the brain's glymphatic system becomes more active, facilitating the clearance of metabolic waste products, including proteins associated with neurodegeneration. Poor sleep quality or insufficient sleep duration can impair this cleaning process, potentially allowing harmful proteins to accumulate over time. The orexin system, which Lemborexant targets, plays a central role in regulating not just sleep and wakefulness, but also other physiological processes including appetite, mood, and stress responses. Dysregulation of this system has been implicated in various disorders beyond insomnia, suggesting that medications targeting orexin receptors might have broader therapeutic applications than initially anticipated. Understanding the intricate relationships between sleep, orexin signaling, and neurodegeneration opens new avenues for both treatment and prevention. It suggests that addressing sleep problems early and effectively might be one of the most accessible ways to reduce the risk of developing neurodegenerative diseases later in life. This perspective shifts sleep from being viewed merely as a symptom or consequence of brain disease to being recognized as a potentially modifiable risk factor and therapeutic target. Implications for healthcare and society. The potential impact of these findings extends beyond individual patient care to broader healthcare system and societal considerations. Neurodegenerative diseases, particularly Alzheimer's disease, represent one of the most significant healthcare challenges facing aging societies worldwide. The economic burden of caring for individuals with dementia is staggering, and this burden is projected to increase dramatically as populations age. A medication that could slow the progression of neurodegeneration, even modestly, could have profound effects on healthcare costs and caregiver burden. By preserving cognitive function and independence for longer periods, such treatments could delay the need for intensive care services and reduce the emotional and financial toll on families. Furthermore, if Lemborexant or similar medications prove effective as preventive agents, they could potentially reduce the overall incidence of neurodegenerative diseases, fundamentally altering the trajectory of the dementia epidemic. The accessibility and tolerability of oral medications like Lemborexant also represent significant advantages over more invasive or expensive treatments. Unlike antibody therapies that require regular infusions and intensive monitoring, an oral sleep medication could be easily incorporated into routine care, making it accessible to a broader patient population. This democratization of treatment options is particularly important given the global nature of the dementia crisis and the need for interventions that can be implemented across diverse healthcare settings. Conclusion: a new chapter in neurodegeneration research. The discovery that Lemborexant can protect against tau-mediated neurodegeneration represents a significant advance in our understanding of the relationship between sleep and brain health. It demonstrates that targeting specific sleep mechanisms can yield benefits beyond simply improving rest, potentially altering the course of devastating neurodegenerative diseases. While important questions remain to be answered, particularly regarding sex differences in treatment response and optimal clinical application, these findings offer genuine hope for new therapeutic approaches. As research continues, we may be witnessing the emergence of a new paradigm in neurodegeneration treatment, one that recognizes sleep as a crucial therapeutic target rather than merely a symptom to be managed. The convergence of sleep medicine and neurodegeneration research exemplified by this study promises to yield additional insights and therapeutic opportunities in the years to come. For the millions of individuals affected by or at risk for neurodegenerative diseases, and for the researchers and clinicians dedicated to helping them, these developments represent a beacon of hope in what has long been a challenging therapeutic landscape. The journey from laboratory discovery to clinical application is often long and complex, but the strong scientific foundation established by this research, combined with the existing approval and safety profile of Lemborexant, suggests that the translation to patient care could proceed more rapidly than typical for entirely novel compounds. As we await the results of human trials, this work serves as a powerful reminder of the importance of supporting basic and translational research, the value of investigating connections between seemingly disparate biological systems, and the potential for existing medications to reveal new therapeutic applications through careful scientific investigation.